and that's known as the tertiary structure. Okay? Now this particular little model here is actually looking at the structure of hemoglobin, the protein that in our blood that binds the heme with an iron in it, and ultimately carries oxygen in it. And hemoglobin floating around in your bloodstream though doesn't have a single polypeptide associated with it. It actually has multiple polypeptides associated with it. And it's the association of these multiple polypeptides that forms the ultimate what's known as the quaternary structure of the protein. And so that in the case of hemoglobin, the particular structure is you have two different polypeptides. It's one that is called the alpha subunit, the other one is called the beta subunit, and you have two copies of each of those subunits. So it's known as an alpha-2, beta-2 structure. So there are two alpha polypeptides and there are two beta polypeptides. And the overall association of these altogether is the quaternary structure. So we'll be going through and talking about what are the stabilizing forces, what's going on with the secondary structure, stabilizing forces and for the tertiary structure, and then also the quaternary structure. All right. Sort of the overview here is one, we have the primary structure is the sequence of amino acids. In a protein. And then the secondary structure localized spatial arrangements of the peptide backbone. The peptide backbone being, the chain of amino acids. We're going to talk about this. This is like the alpha helices and the beta sheets, that kind of thing. Then the tertiary structure is the overall three-dimensional arrangement of a single polypeptide. Finally, the quaternary structure is the spatial arrangement of multiple polypeptides. When we get into quaternary structure, we start getting into the complexity of things. The simplification of sort of that the central dogma is that DNA encodes a protein, and that, it's the DNA is transcribed to make mRNA, and the mRNA is then translated to make a protein. But in fact, there's a lot, as we see, that when we really look at the proteins that are in the cell, most of them are more complicated than just being the amino acids encoded by a single gene in a protein. Most of them have either been extensively post-translationally modified, or they've been the assembly of several different polypeptides encoded by several different genes in there. So we begin to start seeing that complexity in the quaternary structure. All right, so we're going to focus on, first of all, focus on the secondary structure, okay? And to do that, I want to go back, and we talked about this a little bit. We're going to review a little bit the peptide bond structure in geometry. Okay, and so let's build a peptide. Let's see, let's build. What's your first name? K S H A. We'll just do 4. K S H A. Okay. So what is that? Lysine, serine, histidine, alanine. Okay. Okay, so we're going to build the first 4 amino acids of Kashana's first name there, okay? And so let's see, we're going to take our here's our lysine. I'm going to draw the free amino acids first, and then we'll link them up in a peptide. This is review, but this is an important review. So there's our lysine. Serine. Histidine. And alanine. Okay, there's our 4 amino acids, and now we're going to form peptide bonds between these 4 amino acids here. Okay, here's our first peptide bonds. C carbon, double bond O, and then we have the peptide bond to the next nitrogen. Not bad. It should be just CH2OH. Yeah.
In terms of serine and lysine, how do we know that it's always going to be the optimized? Because we can always have like carboxyl process that oxygen on the like how does it always like make something like that reaction cycle?
Oh, I see what you're saying is, yes, why does it link? It's because the actual assembly of this, okay, is done on the ribosome. And it's done using intermediates between tRNAs, actually, that have the amino acids isolated to the tRNAs. And the tRNAs line up, and then you actually form the peptide bond. And so, basically, the enzymes that control that whole process have a very high specificity and structural specificity for the particular amino group in forming the It's not like amino acids floating around in a pot, you're forming amide bonds to them. It's very tightly controlled by enzymes which control the second. All right, so now we have our second peptide bond. And now we make our histidine. And our third peptide bond has the alanine. I would have made life easier if I called on Lady Gaga, right? G-A-G-A, that'd be just life easier. All right. Now here's the things I kind of want to remind you about, is that this amide linkage here, remember that this is really just one resonance structure, that we can also, that lone pair can come down here, and this can come up like this. And so, in fact, this bond has some double bond character, okay? Which means that there isn't, that you can either be in the cis or trans configuration across here. This hydrogen either can be pointing down or can be pointing up, but the way it is in proteins almost all the time is that it's trans, that it's down, like the way that we wrote it. It also means that this hydrogen, this nitrogen, this carbon, and this oxygen are all in the same plane. And we can kind of draw a square around like this, and that's the amide plane. And we can draw another amide plane on the next peptide bond. And we can draw another amide plane around that peptide bond. What this leads to is a better understanding of what the conformational flexibility of this chain is. That basically, this bond here doesn't rotate, so this is all fixed, and this is all fixed. So the only thing we can really relate, we can rotate, is this around this and this bond, and this plane around this and this bond. So we talk about these different rotations using a terminology that describes this bond angle here and this bond angle here. This is known as the phi, and this is known as the psi. Did I get that right? Yeah. Phi and psi. So around each amino acid, remember this is our side chain, that we basically associated with each amino acid is a phi bond angle and a psi bond angle. So for this serine, there's a phi bond angle associated and a psi bond angle associated. For this histidine, there's going to be a phi and there's going to be a psi here. Yeah.
So, you explain how the they're rigid, but the residues or the amino acids, does it matter if they also put them in or they all put them in?
Oh, these guys here?
So would it also be?
No, these can rotate around. These can rotate around. Yeah, as a matter of convenience, I put them all on the top. But we could write them up and we could write them down. And actually, when we start looking at some of those structures, you'll see sometimes they're up and sometimes they're down, and sometimes they alternate. And that's going to be associated, actually, exactly where that's positioned around here is going to depend upon these phi and these psi or essentially where it's positioned defines the phi and the psi bond angle. So we can basically describe the spatial arrangement of this chain just by describing the phi and psi bond angles all along the chain. And then, we say, okay, this bond angle is such, and the next bond angle is such. We could actually map out the whole chain just by recording the phi and the psi bond angle. There is some limitations, though. Depending upon on the bulkiness of the side chains, they can twist around. If you have a really big side chain, I guess the licensing moderately big, the histidine moderately big, there's going to be less conformational flexibility than if you have a lysine here. If you just have a hydrogen, that thing can just spin around, right? And if you have a big bulky side chain, then there's less possible angles that it could actually be on. Similarly, if we have a proline, remember proline is the secondary amino group. The proline has it's fixed, right? Between this carbon and this nitrogen, it's actually fixed. And so there's no flexibility with the proline. So I guess what that leads to is depending upon the sequence of amino acids, you're going to have certain limitations or possible spatial arrangements depending upon the sequence of amino acids. Okay, what you can actually do is plot. I'll draw a real crude one, then I'll show you a picture of one. For all your amino acids, you can plot the psi bond angle versus the phi bond angle. It goes from 0 to 180, here to minus 180, 0 to 180, to minus 180. And when it turns out that because of these limitations, that basically there's an area here that's permitted, that is, that can actually happen. There's an area here that's permitted. There's a little area out here that's permitted. And then there's a little area out here that's permitted. And what I'm basically just saying is that there isn't total free rotation around those bond angles, depending upon the side chain, and they'll tend to cluster at certain bond angles. This plot is known as a Ramachandran plot. Yeah.
So you said you can map out a protein based solely on the phi and psi, but you also said that the side chain could be on either side. So doesn't that give you two possibilities for any phi-psi combination?
No, so where it is around there, actually, so basically and here's where I'm struggling a little bit, is I can't tell you exactly where 0, 0 is, okay? But if it's one direction here or versus one direction here, those are going to be 180 degrees apart, okay? And so where that side chain is as it rotates around, that's what's going to, that's what phi and psi tells you is basically where that is. And, and it goes from positive 180 to minus 180.
Okay.
So here's just sort of another depiction. If here we have our alpha carbon here, here's our plane, so we have this rotation that comes around, and we have the phi and the psi bond angle there. And then, again, just sort of like I drew, is we basically, the chain of amino acids is basically a series of amide planes that, with these side chains, and then this, the phi and the psi bond angle here is established where, the way this one is drawn around, for example, it has the side chain sticking down here, this one up here, down here, up here, down there. And then here's another view of a Ramachandran plot. Okay, and we're going to start talking about secondary structural elements, and what this plot basically shows here is that when a particular amino acid, chain of amino acids are in an alpha helical arrangement, we'll define exactly what this is just a second, this is what the bond angles tend to be. And if it's in a beta sheet configuration, this is what the bond angle here is. This is sort of a strange left-handed alpha helix that shows up occasionally. And down here, I think, are some turns and that sort of thing. But basically, it's in these regions in which we mostly see the bond angle. And when you establish a protein structure, when you get the crystallographic data, you know, there's a deductive process of basically, when you crystallize a protein, you get an X-ray diffraction pattern, and then from that X-ray diffraction pattern, you deduce the three-dimensional structure of the protein. And there's a certain element of, you basically come up with a hypothesis and see if, a structural hypothesis and see if that is consistent with the X-ray diffraction pattern that you actually get. One of the things when you come before you say, okay, this is my structure, is you make a Ramachandran plot out of your proposed structure and make sure you don't have any really weird bond angles. Because if you have some really weird bond angles, you better go back and really double check and make sure that that really is the structure, that it really is consistent with your data, and it's not you get some misinterpretation of it. Okay. So now, actually, let's go in and talk about alpha helices. Although there's actually one more point I just want to get in here, is that glycine provides the most conformational what's the side chain of glycine? Just a hydrogen, right? So it's small. And proline, that's the secondary amino group, provides the least. All right, so now let's look at secondary structure here. And the first one we'll talk about is the alpha helix. This is referred to you in your Watson book. Remember, they were in competition with Linus Pauling, and what Linus Pauling had figured out was the structure of the alpha helix. Okay, and so what happens here is now our chain of amino acids are going to curl around into a right-handed helix. Okay, so let's look at the basic structural features. One that it's right-handed. 3.6 amino acid residues per turn. Remember, once an amino acid has been incorporated into a polypeptide, it's really an amino acid residue. So about 3.6 amino acid residues per turn. So for one complete turn, it takes a little more than, well, just about 3.5 residues to come around. The pitch. Let me see if I can draw this. Here's our helix here. The pitch is this distance here. How far it moves in one turn is 5.4 angstroms per turn. The R groups poke out away from the helical core. Typically, an alpha helix will typically be about, it varies, but it might be around 12 residues long. Sometimes they're up as 20, sometimes they'll be shorter, but on average maybe 12 residues long. A typical protein might have, let's say, 500 amino acids in it. So you can see that there'll be a lot of different helical components within there. If a typical alpha helix is only 12 residues long, a 500 amino acid protein can have a lot of different alpha helical segments. Next is, can be amphipathic. What do I mean by amphipathic? That means, if we look at the chemical characteristics on one side of the R group sticking out of the one side of the helix, it might be different. So you might have, maybe this is leucine and isoleucine and phenylalanine. Maybe you have hydrophobic residues on one side, and you have glutamate, arginine, lysine, and aspartate on the other side, all charged residues. And so you end up having a difference in chemistry between one side or the other, a hydrophobic phase and a polar phase. You can visualize that in what's known as a helical wheel. So here's our helical wheel. Here's the amino acid chain turning around itself, okay? And then the side chains, it's hard to see the side chains, but they're sticking out here. Here's a three-dimensional view that actually shows in the gold here, these are the side chain residues. So this is space filling model. See how the side chains are stuck on the outside. And then if you go back and basically now look to an end view, so now you're looking down the end, you're taking this thing and you're tilting it this way so that you can now look down in the end, and you look at the amino acid side chains that are sticking out, you generate what's known as a helical wheel that looks like this, in which you're, now again, you're looking down at the end of the protein, and you can see the different residues that are sticking outside. So here, this is an example of an amphipathic helix. So here we have valine, that's a hydrophobic amino acid, leucine, leucine, isoleucine, leucine, leucine, isoleucine. But then on this side over here, we've got lysine, serine, that's polar, threonine, that's polar, arginine, that's charged, aspartate, that's charged. We've got a glycine here, that's kind of nothing, and we've got a threonine. So here we've got charged and polar residues, here we have hydrophobic residues. Okay, and that ultimately ends up working the function is, what this does is provide a hydrophobic surface, and that hydrophobic surface ultimately might, say, interact with another hydrophobic surface of another helix and drive the folding of it so that maybe the hydrophobic surfaces of two alpha helices end up coming together and kind of binding it together. Or maybe that maybe this particular protein is a membrane protein and it sits on the surface of a membrane, and so it needs a hydrophobic surface to stick in the hydrophobic component of the membrane. And so these are the sorts of things you can begin to figure out through this sort of analysis. Let me go back to here to make the next point. Now what we want to do is talk about stabilizing forces. How is this helix actually stabilized? And what stabilizes it is a hydrogen bond between, this is an oxygen from a carbonyl carbon that's involved in the peptide bond. Here is an amino nitrogen from the peptide bond 4 amino acids away, okay, that's turned around here, and they form a hydrogen bond between those. And so throughout the whole thing, you actually have a series of hydrogen bonds. I'm going to try to draw this out a little bit more to emphasize this point. But before getting into that, the other thing I want to point about this is the orientation is such that the carbonyl oxygen are always pointed up, at least the way we're looking at it up. We're looking at it this way. And the amino hydrogens are always pointing down. So that if we remember in our peptide bond, so if here we have one peptide bond, and it basically comes around and it connects to another peptide bond here. Okay, here we've got a partial negative charge. Here we have a partial positive charge here, so we can hydrogen bond. Also, because we have a series of partial negative charges going up and partial positive charges going down, the overall helix has a net dipole. In which one end is negative and the other one is positive down here, because of the consistent orientation of these peptide bonds in the negative charge on the upside and the positive charge on that. And this hydrogen bond stabilizes the helix. So ultimately, what kind of makes the helix form is this ability to hydrogen bond to an amino acid that's 4 amino acids away, to the peptide bond of an amino acid 4 amino acids away. So the other stabilizing force in here is that this arrangement, this alpha helical arrangement here, also really maximizes basically the fit inside the core, that you maximize the van der Waals interactions within the interior of the core. So let me just kind of summarize that and make that, emphasize that point, is that beyond these basic structural features, let's see, the last one I'll put in here is that the overall helix has a dipole. And then if we're talking about stabilizing forces, the big one is the eighth finding. That's one. The next one is maximal van der Waals interactions with the force. You know, back at the very beginning, we were talking about, you know, non-covalent bonding and non-covalent interactions. We said, you know, the hydrogen bond is one of the strongest of those non-covalent. So this sort of the main driver. But you also get a little stability just by maximizing the van der Waals interaction. All right, so that's the alpha helix. Now let's look at the beta sheet. That's the other major quite as many Giants hats on today. We gotta get a relief paper. All right, beta sheet. And here we basically have, again, we have the same kind of hydrogen bond stabilization, but it's from different strands. And so, and there's two different ways. Well, let me show you first. We'll talk about anti-parallel first. Sell some space on the top. So I'm going to draw okay, here's a peptide chain. And notice the way I've drawn it is that this is the carboxy terminus down here, or excuse me. Actually, let me go ahead and draw another one here. This is the amino terminal end over here. And I'm just going to continue this on just to make sure this isn't And this is the carboxy terminal end down here. Okay, now, we have this polypeptide chain, and it actually can come back around, okay, and end up interacting There we go. The second strand now, this is the C-terminal end, this is the N-terminal end, and now we have the opportunity to basically form hydrogen bonds in between these two strands. This is anti-parallel because the two strands are actually running in opposite directions. Now what's difficult in drawing it like this is I basically tried to put the amide plane in the plane of the board. The R groups are actually either sticking out at you or sticking back behind the board. Here's another. Each one of these particular sequences here, this is a beta strand. And what you can actually have it just keep weaving back and forth, okay? And you can get a continued, multiple strands to get together. And so the whole thing put together is the beta sheet. Let me show a little bit nicer picture. So here's an anti-parallel beta sheet, in which these red atoms here are the carbonyl oxygen, and the white atoms here are the amino hydrogens. You see the hydrogen bonding that kind of comes all together. And often, the way these are depicted, they're depicting the three-dimensional structure of a protein. They're kind of shown as, this is known as a ribbon diagram, and they're basically shown as these ribbons with an arrow showing the directionality of the peptide chain. You can basically say, okay, here's a strand going in that direction, here's a strand going this direction, here's another strand going in this direction, and this whole thing ends up becoming what's known as a beta sheet. We'll pick up, I don't want to try to start